Hello, my name is Gabriela Imre, and I'm working as a research engineer at the Life Cell Imaging Facility at Karolinska Institutet in Sweden. In this video, I will talk about some important steps in sample preparation for microscopy, the usual problems, and tips in how to avoid them. The learning outcome of this lecture is to give you the best practice for preparing your samples for microscopy. Good sample preparation is the key in getting the best from your microscopy images. In order to acquire images that will answer your scientific question, the samples you are working with needs to be prepared accordingly. Regardless if you are working with tissues, cells, or organoids, there are certain steps which need to be followed, and some of them are critical for adequate microscopy imaging. During this lecture, I will touch upon some important steps in sample preparation for microscopy. For example, how to choose fluorophores and sample carrier, how to seed cells, and where to put the tissue after cutting it, what to pay attention to when fixing your cells, how to get rid of autofluorescence, mounting medium, and how to handle working with it. When deciding which fluorophores to use to label your sample, you must first make sure that their spectra matches the filters available on the microscope you will use to image. It is not uncommon that researchers want to acquire images in exactly the same way as they read in the publication, and often you will be able to do that. But it is not unusual that your microscope does not have exactly the same specifications, and then you need to do some changes in your choice of fluorophore or fluorophores, or if that's not possible, you might need to buy additional filters for your microscope. Watch the bleed-through video found on our website to understand how to check if your fluorophores will work with the filters available on your favorite microscope. Google LCI facility to get to our website, and under LCI videos you will find the bleed-through video. Choose a sample carrier that is adapted to your experiment. There are many types of carriers that you can choose from cover slips of different shapes and sizes and thicknesses, to glass or plastic bottom dishes, multi-well plates, and multi-chamber slides. For multi-well plates and dishes, you can find inserts too, with membranes made of polycarbonate or polystyrenes. Depending on the application, as for example, co-culture studies, transmigration, chemotaxis, their design might differ as well as the pore size. Here I illustrate only a few of the multitude of options available. The manufacturers are constantly designing new carriers to keep up with the needs of the scientific community, so take a look and see what is new out there. A new type of carrier might make your life easier when designing your experiment and imaging. Decide which objective you will use before you prepare your sample. Only use thick plastic bottom dishes if you are sure you will not need high-resolution imaging. Otherwise, use glass bottom multi-well plates, glass bottom dishes, glass bottom multi-well slides, or cover slips. As you might know, the cover slip on your sample or at the bottom of your multi-well plate, dish, or chamber slide is part of the design of the objective. All objectives from all manufacturers are designed for 170 micrometer thick cover slips and with the sample placed directly against the cover slip. Shown here are examples of 40x oil immersion objective lenses from all the major microscope vendors, and every lens in this image is clearly marked with the number 0.17. The graph at the right presents the calculated maximum intensity in the image of a fluorescent bead, which could easily be your sample 2, versus the deviation of the cover slip thickness from the ideal value of 0.17 millimeters. As a function, as objective numerical aperture. The graphs show that image quality varies greatly even with the same cover glass deviation depending on the numerical aperture. For example, with a numerical aperture of 0.95, even a thickness deviation of 0.01 mm results in an image degraded from the ideal by a factor of 1/2. However, a numerical aperture of 0.4 will exert almost no influence on the image, with the same thickness deviation of 0.01 mm. To compensate for such error, well-corrected objectives are equipped with correction collars that will utilize to adjust the spacing of their intermediate lens elements according to the cover slip thickness. All cover slip manufacturers follow the same labeling system. Cover slips labeled as number 1.5 have a thickness range between 160 to 190 micrometers. Cover slip number 1.5H have an even narrower range of thickness, 170 micrometers plus minus 5 micrometers. Number 1.5H cover slips are more expensive. You should use them than doing TURF, super resolution, and then using very high numeric aperture objectives. Do not use any other thickness than number 1.5 or number 1.5H. If you use cover slips with the wrong thickness, you will not be able to get the best focus possible when imaging. The labeling number 1.5 and number 1.5H are valid for glass bottom multiwell plates, dishes, and chamber slides too. It is important that the cover slip you will use in fluorescence microscopy to be extremely clean. Although cover slips look clean, especially when a new box is first opened, they may have a thin film of grease on them that will not allow tissues, culture cells to adhere well, and that may interfere with some processing steps and ultimately with your imaging. Therefore, cover slips should routinely be washed with acid or base solutions to get rid of this thin film. In our facility, we are cleaning our cover slips using hydrochloric acid. I prepare a 1 molar hydrochloric acid solution in a glass beaker in a laminar flow hood. I put the cover slips, which are number 1.5, in that solution, and I leave them in that solution for a couple of hours. After that, I transfer the cover slips into a beaker containing water, and I wash the cover slip thoroughly. After that, I store the cover slip in a beaker containing 70% ethanol. Before using the cover slips, I dry them under the laminar flow hood. You can clean many cover slips at the same time, so it's not a lot of effort to put in. When purchasing multi-well plates, check the height of the skirt at the bottom of your plate. The skirt is the plastic edge that lifts the bottom of the plate so that the glass doesn't touch anything when the plate is on the table. When you want to image the wells at the edge of the plate with a high skirt, the objective might bump into the stage insert. It is dangerous for the objective and the focus will be lost. In many cases, you will then need to exclude the outer wells of your plate, leaving the plate with 60 wells instead of 96. So we suggest not buying multi-well plates with high skirt. In addition, before buying a whole bunch of multi-well plates from a company, ask for a free sample so that you can test the plate quality, such as flatness, leakage, and autofluorescence, especially for plastic bottom plates. Optical plastic dishes and multi-well plates work well for air and water objectives, but some immersion oil melts the plastic within seconds. This destroys the sample and may damage the objective. On top of that, some plastic bottom dishes can be slightly fluorescent and would influence your imaging, especially in the blue and green channels. So if you have a dim cell, it might drown in the background signal coming from autofluorescence. Thus, you might not be able to see it. Here you can see a multi-well plate with plastic bottom touched by the immersion oil. When seeding your cells for imaging, regardless if you use cover slips, dishes, or multi-well plates, you should aim of having an even distribution of your cells in the carrier of your choice. Make sure the cells are well mixed at all times when seeding them by gentle agitation of the tube and mixing every now and again by pipetting. This will assure an even distribution without high local concentration of cells, like for example in the right images, here at the center of the well and here at the edge of the well. This distribution is due to the sudden movements you perform when you move the plate from the bench into the incubator. After the cells attach, treat them gently and avoid harsh pipetting at the well center or the well wall, as illustrated in the image on the left. Here we describe a simple method that will help you on seeding your cells evenly in a 96 well plate. Seed your cells with medium so that it fills less than half of the well. Place the plate on the shelf in the incubator. Place your hand on the shelf 5 centimeters from the edge of the plate. Slide the plate flat on the shelf so that it bumps twice into the edge of your hand. Do the same thing with the other edge of the plate. Do not move the plate until the cells have attached. Most people who cut frozen or paraffin-embedded tissues place them on a thick glass slide after cutting them. Then they add mounting medium and a cover slip and image through the cover slip and mounting medium. This is not the correct way to prepare a sample. As we mentioned before, all objectives from all manufacturers are designed for a sample where the cover slip is 170 micrometer thick and the sample is directly touching the cover slip. When you place your sample on the slide, the mounting medium ends up between the objective and your sample. This means that you introduce light scattering. Scattering will prevent you from getting the best focus, and the amount of light that reaches the objective will be reduced, so the image will be both dimmer and blurrier. Instead, you should place your sample on the cover slip as shown here, so that when you image, the mounting medium is not between the objective and the sample, and doing so you will have the best imaging conditions. It is easy to put section on the cover slip instead of slides. In this short video, you can see how it's done. Cleaning your cover slip will increase adherence of your tissue sample. As you will see in this video, the procedure of laying a paraffin section or cryosection on a cover slip follows exactly the same step as when putting your section on the glass slide. Tape the cover slip on the slide before cutting, not after, so that you can handle the cover slip more easily. When you are done with all steps, you gently remove the masking tape. On the glass slide, you add now mounting medium and flip over the cover slip. Now you have the best imaging conditions. Cytospin your cells on the cover slip. Cytospins were designed to spin cells in suspension on glass slide, and, as we explained earlier, this is not correct when it comes to imaging. Instead, you should cytospin your cells directly on the cover slip. Use 2 small pieces of tape to attach a clean cover slip to a glass slide, and use the slide only to hold the cover slip in the cytospin. In this way, the cells can land on the cover slip. After running the cytospin, remove carefully the tape, flip over the cover slip, and mount the cover slip as usual. Now we will have the best imaging conditions. You will only notice that the focus is not acceptable until you compare the 2 samples side by side, the one with the cells on the slide and the other one with the cells on the cover slip, as shown here. The cells on the top are on the slide, and under those conditions, this was the best focus we could achieve and the best image to acquire. The cells on the bottom are on the cover slip, and you can clearly notice the difference in the quality of the image, sharper and brighter. Do not cover the ends of the slide with the cover slip. When using an inverted microscope, the sample on the left will not be flat when put on the microscope stage insert. That is because one side will rest on the glass slide and the other side on the cover slip, so it won't be flat. Each image will show a focus gradient. When leaving enough space away from the ends of the slide, as shown in the image on the right, the sample will rest on the slide only on both sides, so the focus will be more even across each image. If not imaging cells live, you need to fix them. The most commonly used fixative is paraformaldehyde, but there are other fixatives too, such as methanol, acetone, glutaraldehyde, etc. The choice of fixative should be determined by your application, cell type and tissue you are using, and the recommendation of the antibody manufacturer. If using paraformaldehyde, there are a few things to keep in mind. When fixing by using paraformaldehyde, only use fresh paraformaldehyde, not the one from a bottle which was opened many times, kept at room temperature or on the bench. You can prepare your own PFA solution. Buy the smallest quantity of PFA powder and prepare a working stock solution. Make a 2-time working stock solution, for example 8%, if you want to fix with 4% PFA. Aliquot most in 50 ml Falcon tubes and some in 1 ml tubes and freeze down. Keep a small quantity in the fridge as concentrated working stock, but not more than 1 month. If you buy formaldehyde solution instead of powder, buy the highest concentration possible. As soon as you receive it, aliquot it and store it according to the manufacturer's instructions. When fixing your cells, warm up only the amount of fixative needed and add it to the culture medium, volume to volume, to reach the desired fixative concentration. After 5 minutes at 37 degrees, then you gently rinse the cells with PBS. Be aware that most commercial formaldehyde solutions contain 10 to 15% methanol. Your antibody might not be compatible with it. Fixing cells only links proteins together. It doesn't set the cells in cement. The sample is fragile, For example, do not push on the cover slip when cleaning emulsion oil from your cover slip. Folded cells, filopodia without cell body, or bent filopodia are signs of damage. Here we can see an example of cells fixed with paraformaldehyde, which was not good. One sees that the cells are partially detached and folded on themselves. When fixing the cells with bad PFA, the cells suffer before they are fixed. Look at the cells under a microscope before and after fixing to identify if the problem comes from the fixative. This type of artifacts in cell cultures are almost always solved by using better PFA. Autofluorescence. The major source of the fluorescence background in imaging experiments involving biological samples is actually autofluorescence. So from where does autofluorescence come from? Believe it or not, it can come from the mounting medium you use and mounting your sample. Autofluorescence can also result from the use of aldehyde fixatives such as formaldehyde or glutaraldehyde. And the detected fluorescence has a broad emission range, including blue, green, and red emission. Autofluorescence can come from certain components found in your cell culture media. Another source of autofluorescence can come from common endogenous fluorophores found in the cells and tissues you are imaging, such as NADPH, flavins, lipofuscin, porphyrins, collagen, elastin, etc. These components are generally fluorescent in the green and the yellow portion of the visible spectrum. The most commonly used fluorophores in immunofluorescence assays are the green channel fluorophores, such as fluorescein and Alexa Fluor 488, and natural tissue components can therefore significantly affect the signal-to-noise ratio when these fluorophores are used. Many mounting medium are fluorescent in some channels. For most of them, the autofluorescence develops over time as the medium ages. The most expensive mounting medium have a very short lifetime written on the tube, so they quickly become fluorescent and should then be thrown away. This gets worse when the mounting medium is mistreated by researchers and regularly forgotten at room temperature when it should be in the fridge or in the freezer. Do you think that your mounting medium is not fluorescent? The reality is that you will not notice the problem until you check it. So here is a trick. One quick way to find out if the mounting medium you routinely use is fluorescent or not in your favorite channel is to image a bubble. Use this technique to focus on the edge of one of the bubbles. Take images in all channels with your usual settings. Then increase the brightness and the contrast in the image. If you see the bubble as a dark spot in the fluorescent image, your medium is fluorescent in that channel. Simply changing mounting medium will increase the signal-to-background ratio in all your images. Never use mounting medium containing DAPI, as this will increase background fluorescence. Instead, perform DNA staining as a separate step before mounting. When you are mounting your sample, always remove all PBS to avoid diluting the mounting medium. If you have too much PBS left when adding your mounting medium, it might not harden and you will end up with a mounting medium having the wrong refractive index, and also the surplus of PBS will add to the curing time. If you use hardening mounting medium, allow it to fully harden on a flat surface before imaging, and it takes usually longer than those 5 minutes as sometimes the manufacturer claims. If you use a non-hardening mounting medium, completely seal the cover slip with nail polish and allow the nail polish to dry properly. Otherwise, the mounting medium will leak and the objective will be damaged. With non-hardening mounting medium, one can separate the cover slip from the slide and restain the sample. If one needs to restain, one should not use nail polish. Quite often, at least when you have your cells or tissues on the cover slip, due to all the washing steps during immunostaining and after drying, you will see a kind of white haze on the surface of your cover slip. Those are salt crystals from the PBS, which you should remove, since I am convinced that you do not want to image that. So before imaging, remove salts by cleaning the top of the cover slip with a drop of water. Also, remove old oil before we start imaging. To clean a cover slip that has been imaged with an oil objective, watch this video and use the technique shown to clean filters without rigs. Remember, a dirty cover slip would highly influence your imaging. When imaging live cells, one needs to cover them in cell culture media. Most cell culture media contain components that autofluoresce when excited, thus negatively impacting your signal to background ratio. If your signal is in the green channel and is dim, try replacing an imaging normal culture medium with a culture medium without flavins, vitamin B12, and phenol red. Last but not least, the bottom of some imaging multiwell plates or multi-chamber slides can be made of a thin plastic polymer. These polymers often show a low green autofluorescence, and if your signal is dim from the beginning, it will drown in the autofluorescence signal. Use plates and chambers with a number 1.5 glass cover slip instead. Glass does not fluoresce at all in the wavelength we use in microscopy. Keep your imaging medium in the dark in the fridge. Only warm up what you need. Many biological molecules fluoresce naturally. In tissue, this is often a problem, as the tissue itself can fluoresce in many channels as strongly as any nice antibody staining. Lipofuscin is the most common molecule to give autofluorescence. Autofluorescence sometimes shows up as a general background, but sometimes it looks incredibly specific and shows up in only one cell type. When working with a new tissue, start with imaging unstained tissue to understand in which channel the autofluorescence is a problem. In some cases, simply choosing an antibody labeled in another channel solves the problem. In this panel, we can see images of different unstained tissues with clear signal coming from lipofuscin. How can you get rid of autofluorescence? One way is by chemical treatment, such as sodium black B, peroxide, cupric sulfate in amino acetate solution, sodium borohydrate, ammoniac plus ethanol, and many more. Be aware that although chemical treatment may reduce the intensity of your autofluorescence, it will affect the immunofluorescence labeling too. Also, some treatments will work better in one type of tissue than other, and chemical treatment might require you to compromise between autofluorescence reduction and antigen visualization. There are also non-chemical treatments, UV radiation for example. This treatment takes some time and is often used in combination with other treatments. Another option is to use a spectral detector to separate the autofluorescence spectrum from the spectrum of the fluorophores used to stain your tissue. Check the literature. You do not have to reinvent the wheel. There are plenty of publications already dealing with removal of autofluorescence in your favorite tissue. All you need to do is to adapt it to your sample and staining. Here you can see an example of successful autofluorescence reduction when using sodden black in monkey spinal cord sections. Lower concentration, less than 0.01% of sodden black, provided insufficient reduction of autofluorescent pigments. However, higher concentrations, above 0.1%, give adequate reduction of lipofuscin-like autofluorescence. So here we listed the most important aspects you should pay attention to when preparing your sample for microscopy. Always match the fluorophore with the microscope filters. Choose a sample carrier adapted to your experiment. Use coverslips of thickness specified at number 1.5, meaning a range thickness between 160 to 190 micrometers. Seed cells evenly and not overcrowded. Put the sample on the cover slip, not on the slide. Do not cover the slide's end with the cover slip. Use fresh paraformaldehyde. Remove autofluorescence from your sample. Allow mounting medium to harden or use nail polish to completely seal the cover slip. Use the maximum volume of medium for live cell imaging. Thank you for your attention.